Hello everyone, welcome to Biotech Notebook. This video covers maturation of T lymphocytes in thymus. Before seeing the maturation process, first let's see a small introduction about T lymphocytes. Okay, as we know that T lymphocytes or T cells or white blood cells, there are two types in these T cells. One is T helper cell and T cytotoxic cells. So both these T helper cell and T cytotoxic cells express T cell receptor on its cell surface. Okay, but there is a difference between T helper cell and T cytotoxic cell, that or T helper cells displays only CD4 glycoprotein on its cell surface, and T cytotoxic cell displays only CD8 glycoprotein on its surface. Okay, then these T helper cells recognize antigen in complex with MHC class 2 molecule. Okay, then these T cytotoxic cells recognize antigen in complex with MHC class 1 molecule. MHC molecules are nothing but major histocompatibility complex molecules. Okay, in this there are two types, one is MHC class 1 and MHC class 2. So these T cells can't recognize the antigen as such. Okay, these T cells recognize antigen, only antigen should be in complex with MHC molecule. Then function of your T helper cells is it identifies an infection and activate other immune cells like B cells, T cells and macrophages to fight the infection. Okay, so T helper cells won't eliminate the infection. It won't directly involve the elimination of infection. It just identify the infection and it activate other immune cells to fight the infection. That's why these cells are known as T helper cells. Okay, then the function of T cytotoxic cells, or it kills cells. Cytotoxic, so cyto means cells, toxic means kill. So cytotoxic, in the name itself it implies it kills cells. So these TC cells kill cells which are infected by virus or bacteria, and also it eliminates tumor cells and cancer cells. Origin of this T cell. So this T cell originates from hematopoietic stem cells in the bone marrow. Okay, so the maturation only takes place in thymus, but these T cell precursors are formed in bone marrow from the hematopoietic stem cells. These T cell precursors still retain the capacity which gives rise to multiple hematopoietic cell types. Okay, then these T cell precursors travel via the blood from the bone marrow to thymus for its maturation. Before going to see the maturation of T cell process, let's see a small introduction about thymus. Okay, thymus is also known as primary lymphoid organ because T cell maturation takes place in thymus. Okay, T cells originates in bone marrow, but it gets matured in thymus. Then this thymus is found just above the heart. Okay, then this thymus is largest prior to puberty, and after puberty it begins to shrink. Okay, so in the childhood before puberty your thymus is larger in size and there will be a more formation of T cells in thymus. Okay, after puberty it begins to shrink. The number of T cells production also decreases as we ages. Okay, so in the childhood very good number of T cell is produced in thymus. Then this thymus is a bilobed gland and each lobe is divided into smaller section. It is called as lobules. Okay, then each lobule contains a central medulla and outer cortex. And this thymus is surrounded by a capsule. Next, let's see maturation of T cells in thymus. Okay, immature T cells, also known as thymocytes, which pass through defined developmental stages in specific thymic microenvironment as they mature into functional cells. Okay, so T cell precursors pass through defined developmental stages in thymus to mature into functional T cells. So thymus is a specialized environment where thymic education process takes place. So this is your thymus, and thymic microenvironment or subcapsular cortex, then cortex and cortical medullary junction, that is between cortex and medulla there is a gap that is known as cortical medullary junction. Then the next one is medulla. Okay, so medulla is the inner part of the thymus and cortex is outer part. Then each microenvironment has its own function, and each microenvironment is populated by different stromal cells. So the thymic microenvironments are cortical medullary junction, subcapsular cortex, cortex and medulla. So first let's see cortical medullary junction. T cell precursors enter the thymus from bone marrow. Okay, T cell precursors are originated at bone marrow and it enter the thymus in blood vessels. at the corticomedullary junction. Okay, this junction is between the thymic cortex, that is the outer portion of the organ, and the thymic medulla, the inner portion of the organ. T cell precursor is enter into the thymus at cortical medullary junction. Okay, so it enter as double negative cell. Then mature T cells leave the thymus as they enter via the blood vessels of the corticomedullary junction. So corticomedullary junction is a place where your T cell enter into the thymus and exit from the thymus. Okay, so entry of your T cell precursors into the thymus takes place here, and exit of your mature T cells also through corticomedullary junction. Okay, at this stage, that is at cortical medullary junction, your thymocytes express neither CD4 nor CD8 markers. Okay, it doesn't have any markers on its cell surface. Therefore, these cells are known as double negative cells. The next environment is subcapsular cortex. Double negative cells first travel to the region under the thymic capsule, a region referred to as subcapsular cortex. So this is your double negative cells. So the cells which are in subcapsular cortex are double negative cells. In subcapsular cortex, the process T cell proliferation takes place, and here T cells begin to generate their T cell receptors. T cell receptors are formed by productive recombination of T cell receptor alpha chain and T cell receptor beta chain genes. Okay, so there will be a recombination between alpha chain and beta chain of T cell receptor, and there will be a formation of T cell receptors on the T cell surface. Okay, in subcapsular cortex there will be a T cell proliferation, and there will be a generation of T cell receptor. Then the cells which successfully express T cell receptors begin to express both CD4 and CD8. Okay, then it becomes double positive cells which move to cortex and which populate the cortex. Double negative cells starts to express T cell receptor. Once the T cell receptors are formed on double negative cell, then it starts to express both CD4 and CD8 glycoprotein on its surface, and these cells are known as double positive cells. Okay, so cortex is populated with double positive cells. Next, let's see events takes place at the cortex. Okay, so most 85% or more immature T cells are found at the cortex. Okay. Then the main features of this cortex is it has distinct set of stromal cells known as cortical thymic epithelial cells. The function of these thymic epithelial cells are thymocyte testing. It will test the ability of the T cell receptors to bind with self-MHC peptide complex. This self-MHC peptide complexes are expressed on the surface of thymic stromal cells. Those thymocytes whose T cell receptors bind self-MHC peptide complex with too high affinity are induced to die by apoptosis process. Okay, so there should not be too high affinity between your T cell receptor and self-MHC peptide complex. Okay, those thymocytes that bind self-MHC peptide with an intermediate affinity undergo positive selection, resulting in their survival, maturation, and migration to the thymus. Okay, so how should be the affinity? Affinity should be intermediate. They should not be too high or too low. Only the cells which has intermediate affinity between T cell receptor and self-MHC peptide complex will go for survival, and the remaining cells are die by apoptosis process. Then most thymocytes do not navigate the journey through the thymus successfully. That means the most of the cells die. It is estimated that 95% of thymocytes die in transit. The majority of the cells die because they have too low affinity for the self-antigen MHC combinations that they encounter on the surface of thymic epithelial cell. So it failed to undergo positive selection. So positive selection results in the formation of single positive cells that express either CD4 or CD8. The cells which leave from the cortex are single positive cells. So single positive cells are released from the cortex into the medulla. After positive selection in the cortex, single positive cells migrate to the thymic medulla where they undergo negative selection. Okay, positive selection takes place at the cortex and negative selection takes place at medulla. Okay, so negative selection process ensures that thymocytes that react against self-antigens are eliminated by apoptosis process. Okay, so in negative selection, the cells which react against self-antigens are eliminated. So thereby we can prevent autoimmune diseases. So the cells which won't react with self-antigen only leave the medulla. That cells are known as mature T cells. Then specialized stromal cells called medullary thymic epithelial cells, dendritic cells and macrophages are populated in medulla. These three cells are involved in negative selection process. So these antigen presenting cells and stromal cells present self antigens on their surface to thymocytes, and thymocytes with high affinity T cell receptors for self antigens are eliminated by apoptosis process. Then the mature thymocytes which express only CD4 or CD8 leave the thymus as they entered. Okay, via the blood vessels of the corticomedullary junction. Single positive cells once enter into the medulla, it exposed to the self antigens which is presented by dendritic cells, then macrophages and medullary thymic epithelial cells. Okay, then the cells which cannot recognize self antigens only leave the thymus through cortical medullary junction, and this is your mature T cells. Okay, these mature T cells enter into the periphery lymphoid organs, that is lymph node and spleen. Your T cell precursors are double negative cells, and once they enter into the subcapsular cortex, here your T cell proliferation takes place and there will be a generation of your T cell receptor. Then once T cell receptors formed, then these double negative cells are started to express both CD4 and CD8 glycoprotein on its cell surface, so it becomes double positive cells. Then these double positive cells undergo positive selection in the cortex and it starts to express either CD4 or CD8 glycoprotein on its cell surface. So these cells are known as single positive cells. So the cells which failed in positive selections are eliminated by apoptosis process. Okay, then these single positive cells enter into the medulla where negative selection takes place. So here the single positive cells are exposed to self antigens. These self antigens are presented by antigen presenting cells such as dendritic cell, then macrophage, and also the stromal cells present in the medulla which is known as medullary thymic epithelial cells. Okay, here the cells which can't recognize the self antigen only leave the medulla through cortical medullary junction. These cells are known as mature T cells. These cells express only CD4 or CD8 glycoprotein. Okay, then it enter into the periphery lymphoid organ where it encounters the antigen. Difference between positive selection and negative selection. T cell precursors are originated in bone marrow, then it enter into the thymus as double negative cell, then it becomes double positive cell, then it undergo positive selection. So in positive selection there should be a intermediate affinity between the T cell receptor on the T cell and self MHC peptide complex expressed by this cortical thymic epithelial cells. The remaining cells are induced to die by apoptosis process. So these cells are failed to recognize self MHC peptide complex. Then negative selection. So in negative selection, some cells recognize self antigen which is presented by this dendritic cells and macrophages. So the cells which recognizes the self antigens are eliminated by apoptosis process. Only the cells which won't recognizes the self antigen are released from the thymus into periphery lymphoid organ. Mature T cells once released from the thymus, it enters into peripheral lymphoid organs like lymph node, spleen and other lymphoid tissues. Okay, these organs provide a specialized microenvironment where T cells explore antigens, that is foreign antigens, to fight against the infection. Okay, thank you for watching my videos. If you like my videos, please do like, share and subscribe. Thank you.